Okay, so this question is about regulation of gene expression. And you've got different genes that have not been named. They've just been given letters and different tissues. And we can see that when there's no messenger RNA present in a tissue, so for example, gene H does not have messenger RNA in the liver cells, we would assume that gene H is not necessary for liver function. When we look at question A, it says, based on the data provided, identify which gene is most likely to encode a protein essential for glycolysis and provide reasoning. So there's going to be 2 points associated. Each bold word is a point. Gene G is likely to encode a protein essential for glycolysis. That's 1 point. Why? Because all cell types perform glycolysis. Okay, every single cell on the planet performs glycolysis. So what is glycolysis, right? So I'm going to kind of go off on a tangent a little bit to review that with you. Again, this question is about gene expression, which involves transcription and translation. And they're using messenger RNA presence to determine to what extent is a specific gene, E through J, expressed. Glycolysis is the most fundamental pathway for energy conversion in living cells. It's thought to be the first metabolic pathway on the planet. It is present in every single living organism, prokaryote or eukaryote. It is the definition of how organisms survive energetically. They first use glycolysis, and we still use glycolysis. We just have all the other processes added on. So glycolysis is a very old pathway, and it's universal. Here's just a review of what that looks like in simplified terms. This is a cell with a mitochondria, so this would be a eukaryotic cell. Glycolysis is this whole process that happens in the cytoplasm to yield 2-pyruvate, which then go on and get broken down further in the mitochondria. So evolutionary speaking, all cells do this and gain 2 ATP from glucose. Aerobically respiring cells continue to break down pyruvate in the mitochondria. But basically, glucose splits in half. That's what glycolysis means. Glucose splits in half. It becomes 2-pyruvate. When it does that, overall 2 ATP are made. And 2 hydrogens kind of fall out of the glucose molecule and get picked up by this electron hydrogen carrier, NAD. So this is like the taxi, picks up pieces of glucose that fall off, and this is the overall energy output of glycolysis. Okay, part B. Part B says, gene H mRNA is present in some tissues, but in those tissues, there's not always gene H protein. Provide reasoning. So the question essentially is, how can some cells have lots of mRNA from gene H, but have no protein from gene H? This now is a question about the regulation of gene expression. And when I taught this in class, I used the analogy of a pizza, right? So let's say you're going to, you're home. You're not going to go home, but you've been at home now for 3-4 weeks and you want a pizza. So you go into the freezer, you take it out, you put it in the oven. Well, no, you heat up the oven. You put the pizza in the oven, you let it cook. You take the pizza out, you let it cool a little, you cut it, you slice it. At any moment in time, you could abandon that mission, right? You can speed up, slow down, or totally abandon the pizza cooking. You could even get the pizza out, have it in your hand, get ready to take a bite, and then decide you're throwing it in the trash, okay? So just like that, when we make a protein, okay, so the protein's the pizza. When we make a protein, we have multiple ways of regulating gene expression. What are those? Well, you can regulate how tightly wound this is. You can regulate whether or not the gene is transcribed. You can regulate how fast the gene is transcribed. Then when you get the messenger RNA, you can regulate the 5' cap poly A tail. You can replicate. You can regulate the removal of introns. Then when you get to translation, you could have like not mRNA, but RNAi come in which would block translation. When you get to the protein, you could destroy the protein. So this is like, I take the pizza out of the oven. Sorry, I take the pizza out of the freezer. I put it back in the freezer. This is like, I'm preheating the oven. I'm going to put it in, but I'm going to change how fast or how slow this is, or maybe the toppings, right, Tim? So basically, in the making of a protein, we can regulate the production of the protein at various levels. Here are the mechanisms of regulation listed for you. Number 1, chromatin structure. Euchromatin, heterochromatin. This is the tightly wound, less likely to be expressed. More loosely wound, more likely to be expressed. Transcription factors. General ones, they attach to the Tata box. These are necessary for transcription. Specific transcription factors, these increase or decrease transcription rate. mRNA processing and export out of the nucleus. So this would be removal of introns. And this would be the 5' cap, the poly A tail. RNAi, the little I stands for interference. These are small RNA molecules in the cytoplasm that can bind to messenger RNA and either destroy messenger RNA or block its attachment to a ribosome for translation. And then the last one is protein degradation, which is basically like you have the pizza, you're ready to take a bite, and you throw it in the trash. So as soon as the protein's made, it's hydrolyzed. It's totally broken up and not present at all. So now you're thinking, that's great, Kazishki, but what's the answer to the question? So let's go back to the question real quick. Where did it go? The researcher observed that tissues with high levels of gene H mRNA did not always have gene H protein. Provide reasoning to explain how is it possible to have lots of mRNA for a gene and have no protein. Another way of saying that is, how can some cells have lots of mRNA from gene H but no protein from gene H? So basically I just repeated myself. The answer is, we got to look at ways gene expression is regulated from right here to right here. So something that must be happening between mRNA and protein to get the positive test for the mRNA, but no protein present. So it's not going to be this stuff. It's going to be either one of these. College Board says, for your answer, you could say the mRNA was never exported or exported. That's how you pronounce it, right? The mRNA was never exported out of the nucleus. So perhaps there was an issue with the 5' cap or a poly A tail. College Board says you could also have the mRNA was not translated. And then to elaborate on that, you would say RNAi, or you could just say translation itself was blocked. I'm not sure how much detail they would require. And then the third thing you could say is that there was post-transcriptional modification. So that would come in with this. Post-transcriptional modification that changed the messenger RNA to give it like a different identity. So perhaps in the research, they were only looking at the primary transcript and not the mature messenger RNA. I don't really have this written out neatly, but I'll show you my chicken scratch. So it's either the mRNA was not exported out of the nucleus, the mRNA was never translated, perhaps due to RNAi, the mRNA underwent post-transcriptional modification to change it. And this would be removing of introns. I hope that's helpful. It took a little longer than I thought. We got one more coming up.